this is cybernetics of Albanus. We're going to build a $7 million man.
Some of these marching band. College marching band.
No, it's on DCMB, not DCMB, not DCMB. CMB. So it's next to but they always go by CMB. Washington, D.C. marching man.
I'm still here.
I put your name on there. Glad to know. Glad to know I didn't lie on the sign-in sheet.
Falsifying documentation.
I was thought to get like fired.
12 o'clock.
Sign someone in.
I'll fix that.
We're so close. Is it 12 o'clock? Yeah.
That's
Okay.
Lying in the workplace.
Welcome everyone. We're going to go ahead and get started. Welcome to Tools and Technology Seminar Series. There is a sign-in sheet. I think a lot of you signed in, but if you have missed it, we'll pass it around in a minute. If you already signed in, please just pass it on to the next person. Helps us with the pizza, so I appreciate everyone signing in. So I'm pleased to present today's speaker. We have Ricardo Albanus, and he is in Steve Parker's lab.
All right, thank you. Thank you, Marcy, for the introduction. It's glad to be here at Tools and Tech again. So, as Marcy said, my name is Ricardo Albanus. I'm a fifth year bioinformatics PhD student at Steve Parker's lab. And today I'm going to talk about a method we developed at Steve's lab to predict transcription factor binding from ATAC-seq. So, I want to start by reinforcing why we should be interested in transcription factor binding in the first place. So we know that, especially in the case of complex diseases such as type 2 diabetes, the majority of the variants that we detect with genome-wide association studies are non-coding. So this means that rather than altering protein structure, these variants are affecting regulatory circuits in the cell, which in turn leads to increased disease predisposition. And we have evidence that these variants are affecting transcription factor binding by either creating or disrupting binding sites. And this leads to downstream changes in gene expression. So to illustrate the concept, on the right here we have this example of an A to C variant, right? So there's a transcription factor binding site here. So the transcription factor regulates these genes downstream, and you can see that the A, you have a binding site here, so the binding site is intact, but whereas with the C, this disrupts the binding site. So you can imagine this could be like an insulin gene or some pathway, relate some important pathway for the disease. So this means that to understand what this non-coding regulatory circuits are doing, we need to know what is happening at the transcription factor level. So if you know which transcription factors you're looking for ahead of time, you can do a chip-seq experiment, but you're limited to one transcription factor at a time. You have to sequence every time. So one alternative, if you don't know what transcription factor you're looking for, or if you have multiple transcription factors that you want to look at the same time, is to predict transcription factor binding from chromatin accessibility profiles, which we can generate from assays such as ATAC-seq and DNA-seq. And ATAC-seq is a very versatile assay, which is using the TN5 transposase to integrate the genome at accessible regions and generate these open chromatin profiles. And to determine where the TFs are binding, we can look at the cut patterns in the ATAC-seq data. And you can see on the schematic here, we'd expect, can you see it? No. So we'd expect low cut densities where the TF is sitting because it's protecting DNA cleavage, right? And we call these transcription factor footprints. But turns out that not all the transcription factors leave footprints. And this is a figure from Gordon Hager's lab. And it's showing aggregate DNA struct profiles over transcription factor bound motifs. You can see the motif here at the center. And this first plot here is showing CTCF, and you can see that it leaves a very deep protection. So the amount of protection here from cleavage compared to the vicinity is higher, right? I'm highlighting this with a black box here. But that's not the case for all the transcription factors. I have examples here of AT1 and glucocorticoid receptor, and you can see particularly glucocorticoid receptor at the right leaves a very faint protection, right? So it was hypothesized that the level of this DNA protection is a function of the time that the protein is binding to DNA, its residency time. And turns out that CTCF and AP1 have long experimentally measured residency time, whereas the glucocorticoid receptor is known to bind very transiently, in less than a second for each binding event. So the way we know this transcription factor residency times, or at least some idea of how long they're binding to the DNA, is by using FRAP assays. So basically you transfect your cell with a GFP tagged version of your protein of interest. Then you bleach part, photo bleach part of the nucleus, just shining a laser there. And then you count the time that it takes for the fluorescence to recover. So that means the molecules that are outside the bleached area are going into the bleached area. Right? And this recovery time will be a proxy of the transcription factor DNA dynamics. So freely diffusing or highly mobile TFs will recover very shortly, whereas slow-moving TFs will take minutes and maybe hours. If you're doing like a histone FRAP recovery time, it literally takes hours for those to recover because they're so non-mobile that it takes time. So, but the problem with those experiments is that they're very, very hard to perform. So there's only data available for a handful of transcription factors, like around the order of 20 to 30 transcription factors. And there's actually a ton of interesting and relevant biology regarding transcription factor residency times, but because focusing more on the tools and tech aspect here, I'll focus on the question that's directly relevant to us here. So how do we know which factors leave footprints and which don't? So that would be a function of the residency time. Yes?
This case, you measure recovery time. It's not the same as residence.
Yes, so it's a proxy.
You measure the mobility. For example, even if a protein resides on its binding site for a very short amount of time, but the binding site is very frequently happening around the chromatin, you will still observe that the mobility is quite limited.
Sorry. Yeah, I see what you're saying. And there's another type of experiment where they do a single molecule tracking. So you're doing it at the single molecule level. And there's high correlation between the single molecule times and the FRAP recovery time. So the FRAP is a population measure, right? You have your entire population of proteins with the GFP tag. But still, we know from non-biology, for instance, histones will take hours to recover. CTCF and cohesin also takes many minutes. And these factors like glucocorticoid receptor, where you know that they bind very transiently, they recover very fast. So it's the FRAP recovery time, there's paper, there's literature showing, it's an upper bound of the transcription factor residency time. So this leads to the main topics that I'm going to cover during this presentation. So first is how to use the ATAC-seq data to infer these dynamics of transcription factor DNA interactions, and more importantly, how much are the TF binding predictions influenced by these kinetics. And then I'm going to talk about our methods to predict transcription factor binding, not depending on finding footprints. Right? So you might have seen these slides before, so if you have, go ahead and take some pizza. But if you haven't, this is an important concept. So keeping what I said in mind about residence time and TF-DNA interactions, let's do a thought experiment where we have of what we would expect to see in terms of chromatin organization for a transcription factor that binds for a very long time in DNA, something like CTCF is known to do. So it will phase the local nucleosomes around. So if the nucleosomes are phased around the TF, we're going to have this evenly spaced shaded regions where we would expect low PN5 integration probability, right? So they're protected from cleavage. Then we can add the y-axis corresponding to the ATAC-seq fragment size and see what is the expected fragment distribution that we would see in this case. So first, the small fragments would be adjacent to the transcription factor binding sites. We'll color them in blue if they're to the left, or in black if they're to the right. Then we'll see some slightly larger fragments integrating on both sides of the transcription factor. We'll color them in red. Then we'll see some nucleosome size fragments, so they're integrating on both ends of the adjacent nucleosomes. You have dinucleosome size, so they're spanning two nucleosomes, and so forth. Right, and instead of showing the actual fragment, I just show the fragment midpoint, so where is the middle of that fragment. And in the end, we'll have these clouds of fragment midpoints forming these V patterns around the transcription factor binding site. So Steve Henikoff looked at those in the context of MNA-seq data, and we're looking at ATAC-seq, so it's a different application. And now let's see what does a real V-plot look like. So on the left, we did a V-plot on CTCF-bound motifs on GM2878 data. Again, the x-axis is the distance to the motif center, so the motifs here right at 0. Right? And the y-axis is the ATAC-seq fragment size. And each point here is going to be a different fragment midpoint. And the projector is good enough that you can see these clouds of fragment midpoints forming this V pattern around the CTCF binding site. Then we can ask, will we see this for every transcription factor? So if we look at NF-kappa B bound motifs, again using ATAC-seq data, we don't see anywhere close to that pattern. So, and I would like to say that these plots have the same number of points going into them. So it's not that you have more signal on CTCF versus NF-kappa B. It's the same number of points. It's all about the organization, the underlying chromatin architecture. Right? So we can quantify these differences. So let's look at the zoom in on that CTCF plot. And we use an information theory approach to measure how ordered a given V-plot is. So we can do this by calculating the information content of the fragment size distribution, which will tell at every position in the x-axis whether the points are uniformly spread along the y-axis or if they're concentrated. So let's look at the example right here at the center. We have high information here because the majority of the fragments are small fragments next to the binding site, right? So if this is high information. If it was low information, then basically we'd see a uniform distribution of these midpoints along the y-axis here. So I put this information content track here. You can see it's a tall arrow. If we move a little bit to the left, we see there's still information. So you see this cloud of midpoints here, but there's also this other cloud. So it's a little bit more dispersed. The information is high, but not as high. Then if we move to this part of the V-plot, we have this first nucleosome size fragment, so it's the first nucleosomes around this position, and the information goes up. And we do this at every position until we reach the end here where we have low information. We also do it on the other side. So we have this track of the observed information for this V-plot. And then we can just randomly shuffle the points here, and we get a node distribution. Right? So if we divide one by the other, we get this information content enrichment. And we can focus at the center and side peaks here, which are a fraction of the transcription factor binding and the adjacent nucleosomes. And we can calculate what we call the feature V-plot information content enrichment, short for FVICE for short. And this is giving a single number, a quantitative measurement of the impact of the transcription factor in the local chromatin environment. Right? And we can do this for every transcription factor if we know where they bind. Right? So looking back at those CTCF and NF-κB plots, you can see that there's a 5-fold difference in the V-plot information content between the two. So CTCF is much more organized than NF-κB. And again, this is the same number of points that we're looking for each plot. Right? So, and note that I'm normalizing to the CTCF value. So CTCF is 1. And this is how it's going to be for the next plots. So we did that for all the factors that we have ChIP-seq data available for GM2878. And you can see this range of V-plot information contents from 0, pretty much no information, all the way to 1, which is CTCF. And CTCF and AP1, those guys with the deep footprints, they have the highest scores in this group, right? So if the V-plot information content correlates with footprint depth, then basically everything that's to the left of CTCF and AP1 won't leave very deep footprints, and we probably do a poor job of predicting where they bind. So based on what I just shown before, it seems like using footprints to predict TF binding can be potentially problematic for the majority of the transcription factors. So we need to come up with a method that does not depend on finding these footprints or any kind of modeling of the ATAC-seq signal. Right? And ideally, we should use robust metrics that should be hopefully less affected by factors such as signal-to-noise rate or sequencing depth. Right? And our attempt to solving those issues is called Nemo, and it's a count-based algorithm that's looking at the ATAC-seq signal at the vicinity of motifs, and also at the number of co-occurring motifs. Right? So both of these metrics were shown to positively correlate with transcription factor occupancy. So basically, the more accessible your motif is, the more likely the TF is going to randomly encounter it when it's diffusing along the nucleus. And the more binding sites, the more chances of your transition factor dwelling on that region. So a way to think of that, imagine like a bee, your transition factor is a bee, and the binding sites are flowers. Right? So if you have one big open flower, it's more likely that a bee is going to bind there compared to a closed flower. And if you have multiple flowers, it's more likely that the bee is going to be around on that region compared to like a bush that only has one flower. Right? So that's the general idea behind our modeling. And like I mentioned, Nemo is a count-based model. So this means that for each transcription factor, so it's a per TF approach, we model those two distributions using two different negative binomials, one for the ATAC-seq signal and one for the co-occurring motifs. Right? And on the right here, I'm just showing an example fit here for ELF1. That's a transcription factor that's fairly easy to predict. And if we were to predict ELF1 solely on accessibility, so we have this on red is the model distribution, on black is the observed data. We have on the x-axis the number of ATAC-seq fragments in the vicinity, so you can see that the majority of the data is less than 20 fragments. So ATAC-seq signal is less than 20, right? And let's say if we want to predict binding just on ATAC-seq signal, we say, let's say, we want an false discovery rate of 5%, and we say everything that's to the right of the false discovery line we predict as bound. So you have to have at least 24 fragments. Every ELF1 motif instance that has more than 24 fragments, we consider that as bound. So now let's look at how we obtain these model distributions, both for the ATAC-seq signal and co-occurring motifs, and also for the how to get a single p-value out of two distributions. So for the ATAC-seq signal, we calculate the number of ATAC fragments in the vicinity of every motif instance. So for every motif, look plus minus 100 base pairs, count the number of ATAC-seq fragments. We ignore fragments that integrate directly at a motif sequence. So it's been known that both for DNAs and ATAC-seq, that they have specific sequence bias. So it's not completely random how it's cutting DNA. And if you imagine every motif instance here, we're going to have like the motifs at the center and this plus minus 100 base pair on all of them. the motif sequence is going to be basically the same across all of those regions, right? So we want to avoid any kind of integration bias and just ignore everything that's integrated directly at the motif. So it's a Urban version of dealing with the ATAC-seq bias. So we fit a negative binomial distribution on the motif instances outside ATAC-seq peaks, right? So the vast majority of motifs outside ATAC-seq peaks will be unbound, right? It's closed chromatin. And this is going to be our null distribution where we calculate the p-values for all the motif instances. And for the co-occurring motif, it's more simple. We just fit a negative binomial distribution on the number of neighboring motifs. So how many other CTCF sites are next to CTCFs, that CTCF instance, or how many ELF1 instances are next to the one that you're looking for. And then we fit a negative binomial on that, and again, we can calculate a p-value. So let's say if you have, for CTCF specifically, you have one CTCF with 5 other CTCFs next to it, that's going to be highly significant. So we take the p-values from the two distributions and combine using this LibTech method, which is basically just convert the p-value into a z-score and then sum the z-scores. Right? And we define bound instances using, instead of a false discovery rate, we use the Benjamini-Iecucelli correction. So this is a little bit more stringent than the regular Benjamini-Hochberg because it's modeling the dependencies of the data in a more general way. So this is ideally suited for cases where you have a low proportion of true positives in the data. So you expect to have few binding sites, like out of the 300,000 or 500,000 CTCF motif instances, you expect like 20,000 of them to be bound. So that's why you use this. Okay, so let's compare. So we wanted to see how BMO compared to different methods. So we compared first to Centipede from Roger Picciarelli. He's now at Wayne State, but this was developed at Richard's lab. This one is not directly modeling footprints, but it's modeling the ATAC-seq signal at the vicinity of each binding site. We have DNA SUTF from Gordon Hager's lab and Hint from the Costa lab. Both of these are doing footprint predictions and also some ATAC-seq bias modeling, actually DNA-seq bias modeling. Then we have PEAK, which is also a footprinting method from David Gifford's lab. But this one is grayed out because it does its own internal motif scanning, and it's kind of hard for us to directly compare the two methods. So I can show data later if you want. And our baseline is just web tools intersect motifs with ATAC-seq peaks, and this is the most simple way of every motif instance that's within an ATAC-seq peak, we consider that as bound. So let's show the comparisons. First, we're going to look at precision recall AUCs. We're comparing this to the real ChIP-seq data for ENCODE and see how well those methods predict the real binding site from ChIP-seq. Right. So for all these plots, in the next couple of slides, I'm going to be showing BMO on the y-axis, so that's our method, and the other methods on the x-axis. Right. So each point here is a different transcription factor. You have the key here. And if a point lands above the diagonal, it means that BMO is better for that transcription factor in that method, right? And I have here the total experience, so 37 ChIP-seq experience, and the number of points falling above the diagonal, right? So if we look at Centipede, we outperform Centipede 3/4 of the time. Then we can look at DNA, SUTF, and Hint, and basically outperform them on this precision recall AUC metric. This is when rostro data, so this is one ATAC-seq data set, but we also have ours. And this is what we see on our data set, so very similar. But I don't want to focus on precision recall AUCs. I want to focus on F1 scores. So if you're doing this kind of method prediction, it's not enough that if you rank your predictions by their score, that you can separate between the bound and unbound classes. You want to test if the threshold that your method gives, so say more than 24 reads are predicted as bound, is this better compared to the other methods. So taking into account both the precision and recall as well as the cutoff for each method. So this, I think, is a more realistic way of comparing those methods. And again, you can see we outperform Centipede, we outperform the other methods. This is the when-roasted data. And again, for our own ATAC-seq data. So
here, for each different point, are you using the same threshold or TF, the secret threshold?
So for our method, yes, we model each transcription factor separately, and then DNase to TF, and hint, it's going to give you a bad file of where there are footprints, and you have to intersect that with the motif instances to figure out, because it doesn't tell you which transcription factor is binding there. It just say, I found a footprint there. So you have to intersect that.
So in other words, BMO needs to pre-train all the cutoffs.
No, it's not pre-trained. This is unsupervised.
No, what is the threshold for considering there's the PF binding? You need to pre-train it.
No. We use
Otherwise,
the Benjamini-Yacuccelli correction. It's a false discovery rate. It's a p-value. Nemo gives a p-value.
Yeah.
There's no training.
But here, when you get your F1 score, you need to have a cutoff, right?
Yes. And that's the false discovery, the Benjamini-Yacuccelli cutoff. It's a p-value.
And that cutoff is
Unsupervised.
whatever supervised or unsupervised. Is that automatically learned, or you set it manually, or?
No, that's what I'm saying. We combine the p-values of the two fitted distributions, right? This is all unsupervised. At no point BMO is looking at real binding sites. This is just from the ATAC-seq data. Then we get this combined p-value. We do a Benjamini-Yacuccelli correction, and everything that's less than 0.05 we consider as bound. It's just a p-value.
So
I think just a few minutes back you were talking about thresholds.
Yes, and that's our threshold here.
So
Okay.
for all your points, your threshold is p-value 0.05 in
Yes.
your F1 score plot.
Yes.
So that means there's all transcription factor use the same threshold.
So this threshold is going to be different. So let's say for F1, let's say it's 24 fragments here, right? But for CTCF, we're going to model this distribution differently, right? Because it's modeling per transcription factor what is the distribution. Okay. We can talk later. So another way to look at this data is trying to correlate with those vplot information contents that we looked at before. So on the top here, I have the transcription factors, so the ones with ChIP-seq data, the ones that we are making the predictions, and their associated vplot information content. And I'm splitting them into three groups, so low, medium, and high vplot information. Right? And you can see the overall prediction performance, so this is just the F1 score sum for each of those methods regarding those different vplot information categories. And you can see that Nemo clearly outperforms the other methods, and it's mostly because of these lighter colors here, the medium and the low. So those guys here, they're probably not leaving footprints, and the other methods have a hard time predicting where they're binding. Because if you look at the red bars here, there's actually not a lot of difference between the methods. It's mostly on those two here. And another way to look at this is showing a fraction of the best performing prediction. So the best prediction performance. So for each transcription factor here, whatever method has the best at one score is going to be to the top of this distribution, and then we are going to show each of the other methods as a fraction of the best performing score, if that makes sense. So let's say for AP1, Centipede was the best here. So the second best, so what is the fraction of the Centipede score the other methods have? So if one method have an F1 score of 1 and the other was 0.8, then it's going to be 0.8 on this. Right? So, and hopefully you can appreciate that the more to the left we go, so those low information FYs, the worse the other methods get. Right? And Nemo is always trailing here at the top, and the only exception is CTCF. Right? So now we should ask if most factors are like CTCF, where the other methods are better, or if the majority of transition factors are like everything else in this plot here, where BMO outperforms. So using BMO, we made predictions and calculated the V-plot information score for all the transition factors that we have motifs available. And we can see that basically CTCF is the very tail of the distribution, right? So the majority of the transcription factors are more like the glucocorticoid receptor, which doesn't leave footprints, and not like CTCF. So we shouldn't be using footprinting methods to predict them. And then, just for making things interesting, we fitted a mixture model in this data just to try to estimate what's the proportion of this high versus low V-plot information TFs. And you can see that the CTCF and the high information, so the highly ordered V-plots TFs, account for around like 20 to 25, 26%, depending on the data set of the data. And this is actually strikingly consistent with what Gordon Hager's group, they measured the depth of the footprints, and they found that the factors with deep footprints correspond to 20% of all the transition factors that they measure. So it's encouraging to see this, and these are completely different metrics, right? This is measuring footprint prediction, and we are measuring the V-plotting from the ATAC-seq information content. And I won't go into this in depth, it's hard to see, but we looked for the proteins that are associated with each of those transition factors and look at what are the protein domains that associate with that high and low FIs distribution. And there's difference. So we see different protein domains are enriched for the high FIs, the high information content TFs compared to the others. So this again supports the idea that this might be two different classes of transcription factor. Right? And so let's go back to the photo bleaching assay, and let's look at how combine BMO predictions and look at how they correlate with those FRAP recovery times. So get an idea of if this V-plot information content that we measured correlates with the transcription factor DNA dynamics. So those plots I'm showing here, on the y-axis I have the FIs, so the V-plot information content. And on the x-axis, I have the experimentally measured FRAP recovery time for these factors over here. And hopefully you can appreciate that there's a nice correlation now. So CTCF, guy with the very high V-plot information right at the top here. Then we have PrEP3, it's an intermediate factor at the center here. Right, and then F-kappa B, which you looked before, right at the bottom here. And we did this in our own data set, and we find basically the same correlation. Alex?
So why do you have less different factors here? Is it because of the graph data is not available?
Exactly. Exactly. You don't have for everything. That's the limitation of those graphs, because otherwise we would know. Yeah, so that's something that we're excited to be able to at least give some. Of course, I wouldn't count like the FIs as a direct math, like as a prediction of the residence time, but at least you can get some ballpark if your factor is more like CTCF or if it's more like the glucocorticoid receptor. So without doing those very intensive experiments, right? So before we go to the live demo, we can conclude from this data that I showed here that transcription factors vary in their impact in the local chromatin architecture, and this correlates with measures of residency time. That's what I just showed here. And the negative binomial modeling of BMO is a better overall predictor of transcription factor binding than footprint, and most likely because the majority of the transcription factors have much lower V-plots, so much lower chromatin information than CTCF. So let's do some demo. If you want to use BMO in your own lab, I need to access the presentation. Okay. All right, so you can go. Just move our terminal here. Okay, so you can go to github.com, Parker Lab, Parker Lab-/BMO. Right? So what do you need to use BMO? You just need your ATAC-seq. You need your BAM files, you need your peak calls, and you need one bed file per motif, right? So we have some hints of how to format and get those. All right, and if you want to install, just git clone BMO. I already did that here. And as of now, BMO is built on SnakeMake. So I'll be giving a talk on SnakeMake on November 30th. I'm going to showcase that later. And to make things more reproducible, instead of you having to install yourself SnakeMake and all the dependencies, we have a Conda environment that you can source and activate directly. And this will make it easier if you're in a cluster and you don't have administrative access or simply don't want to mess around with your dependencies, right? So just run this step here, this command here, then you activate. So activating here. And for the first time you do that, you also have to install some of the other dependencies that we cannot include them with Conda, but it's just 2 or 3 packages. And in order to so what do you need to run? Let's show the example here. So you have your bed files here with the motifs. You have your BAM files, and you need the peak file, this motif. This is just the name of the motifs you want to run in case you don't want to run the entire data. And let's look at the config file. You need to make this config file that what SnakeMake uses. So basically you just need to say where is BMO installed, where did you clone, where, like what folder are your motifs, what is the extension of your motifs, the file that contains the motif names, and where do you want to put the results. And then we have some more advanced options. And then for each sample, you just need to say what is the BAM file and what is the peak file. So I have the description of those configs here. And then to run, basically you just activate the environment and run this command here. So SnakeMake is calling it SnakeMake. I'm saying we are running with 4 threads and then giving the config file. And that's it. So now, first it's going to calculate the ATAC-seq signal on the bad file. As you can see, it's crashing my CPU over here. It's going to take like 10 seconds to do this example. And then, so here are all the steps. So first is measuring the ATAC-seq signal, is looking for the second motif. Then it's going to fit the negative binomials, and then finally run BMO. And it's actually already doing that. So now it's feeding the negative binomials. And once this is done, go back here. As this goes, we created this work directory here. that's where the results are coming. And of course, this is taking a little bit longer because it's a live demo. Normally it takes less than that. I was expecting this to happen, but yeah. So now it's reading. This is BMO. So it's just reading the data, feeding the co-occurring motif distribution, calculating the p-values using that sum z-score, and all done. And the next one should be all done here too. All done. Writing output, all done. Yeah, that's it. So we can go to work, BMO, example 1. So it's going to have one folder per file. And in this bound directory, you have the predicted calls, predicted bound, CTCI motif. So everything that's on this bad file is what BMO predicted as bound. And then you can directly use this as input for wherever you want. And if you're not satisfied with our thresholds, we provide this other folder here. It's called all. And this actually has all the matters. So what was the ATAC-seq signal for each motif instance, the number of co-occurring motifs, p-values for the ATAC-seq signal, for the co-occurring motifs, the combined value, p-value, the adjusted p-value. I don't remember what this 0 here is. And then this is predicted bound or not. Yeah, so everything is here. So hopefully I convince you that this is easier to use than everything else that's out there. Oh. Okay. And so I briefly mentioned SnakeMake, but I have an upcoming SnakeMake talk that Marcus was kind enough to invite me for the Pi Live. This is going to be November 30th at 3:30 p.m. So reasons to come. There's free pizza as well, and there will be Python. So with that, I'll take questions. Thank you.
So the input file is from the ATAC-seq data.
Mm-hmm. Yes. Yes, that's the. From the ATAC-seq experiment, you only need the BAM file with the actual reads and the peak calls, right? So because you need to fit the model with the reads outside peaks, right? So it needs that file here, and then one bad file for it.
So at the beginning, talk about the GBars results. Let's see the most variant reported and non-coding variants.
Mm-hmm.
How that can be related to your tool, like that.
Sure.
So your tools will also take some information from that. Or just
So you can do, for instance, you can use Grabber, that ones that try to do enrichment of features in GWAS SNPs. Right? So I actually don't, as a video, I don't have the slide here, but I have a slide. I've done an analysis where we inputted BMO predictions, right? So everything that BMO considers a footprint versus just ATAC-seq peaks. And we get a better enrichment in GWAS hits on the BMO predictions compared to the ATAC-seq peaks. So we're narrowing down on where those variants are. Right? And another way to look at it is like imagine you have big linkage disequilibrium block and you have a lead SNP, that the one that with the highest signal, but you don't really know for all those variants in LD with that lead SNP which of those are which of those are the causal variants, or at least the potentially causal variants. So you can intersect that with the BMO predictions and see like which of those you might want to look further.
So far you don't have a parameter or option can take that information. Do you?
That's up to you, right? So we just generate the predictions, and then however you want to analyze. So if you want to input them regularly, you could, for instance, we did a similar work for Arushi's CNS paper, where we made predictions with Centipede, and then we ran the entire T2D type 2 diabetes catalog on those predictions, and we find that there's this factor called RFX6, potentially, that's enriched for the type 2 diabetes SNP. So it could be a factor that's driving the T2D phenotype. So in that case, we look at every possible prediction and calculated which was associated with disease. So there's multiple ways to look at this there, or maybe you just want to see, is your transcription factor binding at known enhancers of whatever process you're interested in. So you have a pathway, you have the genes, and you have the promoters or the enhancers associated with each of those genes, and you can just see which factors are enriched, are predicted bound, like are enriched there. That makes sense.
So for the required input, the metaxic peaks, they're also called from the BAM files, right?
Yes.
So is there any way you can internalize this step so you only input the BAM file? That will make it much easier.
Sure. Actually, yeah, this is just going to be another snake.
Right.
Yeah, I can make this automatic, so if you don't give a BAM file, peaks file, it's going to call, but then it's going to use max 2. So.
How do you distinguish TF binding and other non-specific protein bindings? Say, I don't know, other structural proteins, non-histone structural proteins bind the same place, so you still call it a peak.
No, you don't have the ability to do that. What you have the motifs, right, associated with your transition factor. So yeah, that's an important question. We're limited to things, to transition factors that have known motifs. You cannot look at you cannot look at factors like, you cannot predict histone binding or anything like that because they don't have motifs. So you're constrained within this motif realm.
Any other questions? Great, thank you.
So we're asking about the threshold.